Is it live?
Yeah.
Oh, you're live.
Oh, I see it now.
Wait. Is that you on?
Yeah.
Okay. That's the one part. Oh, there's a. Hey guys, make sure to. Well, actually, this is going to be an exam review.
Yeah.
So make sure to sign in. The sign in is going to be in the description below. And then also, since it's an exam review, we're going to be doing questions only. So I won't be teaching anything, but any questions you have, shoot away. Just put them in the comment section below or on the side. Anything, literally anything. I brought my Campbell textbook so we can go over anything you guys are struggling with. So yeah. Yeah. So I'll give you guys a second to come up with any questions. Yes, for those that are just signing in right now, make sure to go to the link below and sign in for CSS. And then because it's an exam review, we're going to be doing Q&A. So whatever questions you guys have, shoot away, put it in the comments this way. Literally anything since the very first day till maybe this Friday. Anything you guys have questions about. Yeah, I'm just looking through the textbook to see maybe some certain topics that you guys might have struggled with, but this is going to be mostly student led. So yeah, I'll give you guys a couple of seconds to come up with questions. Yeah, so can review. Yeah, we can do. We'll do the bonds interactions in different protein structures first, and then we'll go to photosynthesis. Let's see. All right, so. Sorry. So. Oh, you can't see that, can you?
Yeah, it's alright. That's better. Hold on, let me turn the shade on.
Oh,
Is this one over there?
So when it comes to primary structure, be, the way you can kind of remember it is that primary is first, and so before you can start getting into these very complex protein structures, you have to create the main chain first. And so the main chain is going to be comprised of. Oh, the thing you need to remember is that proteins are a polymer of amino acids, and so what that means is that amino acids are monomers that are joined together to create a protein because that's a polymer of amino acids. So for the first one, we're going to have, let's say, like a random amino acid connected to another amino acid with a different R group or same R group, doesn't matter. And then another amino acid with this R group. And so the bond between this, at primary level, we're only looking at the main chain. So really, we're only looking at the only bonds that we're concerned about are these right here between the amino acids. These two, yeah, those two, yes, those, and those are called peptide bonds.
Do you want to erase this circle when you circle the peptide bonds because it looks really confusing? Yeah, it looks like a bunch of just
Circles.
yeah.
Let's show it.
Yeah, there you
Peptide bonds, also referred to as a polypeptide. So, and because we know proteins are made of amino acids, then we know that the polypeptide refers to poly being poly meaning poly meaning many, and then peptide just referring to the type of bond. And so basically, protein, when you refer to it as a polypeptide, is just saying, oh, it's a thing made out of many different or many peptide bonds. And so that's a way you can kind of remember the kind of bond that we're looking at in the primary level. Now let's move on to secondary. If this is Ksenia, if this is like the kind of things you're talking about, then give me a thumbs up. But if this isn't answering your question, let me know in the comments. But now we can move on to secondary. Or maybe it should be.
go. Loop it around. This pin particle.
Pin particle? Shut up. Or but
Your data sheet is not super clear. Some marker.
is that clear?
Yeah, you.
Okay. Yeah. Ancient. So when we're looking at the secondary level, we're looking at, all right, cool. So when we're looking at the secondary level, we're still talking about the interactions between the main chain. And so basically, when we're looking at secondary, now that we created the polypeptide bonds and we have a main chain to work with, now the main chain is going to be attracted to other parts of the main chain because of the kind of groups that it has. If you guys remember, an amino acid is made out of at 7.2 pH, the amino acid is going to have a carboxylate. Carboxylate? No.
Yeah.
Carboxylic acid? No, it's carboxylate. Yeah.
Carboxylate.
A carboxylate, that means that it's
Negative.
negatively charged, and an amino group that is positively charged. And so those, even though those are going to be connected to other polypeptides, you're still going to have these kind of groups attracted to these groups of other amino acids. And so that's why you start seeing these patterns of alpha helices where they curl up on each other like this, kind of like DNA where it has a helix to it. That's one of the types of structures it can create, or it can also interact with the main chain can interact with other components by creating a beta deleted sheet. And so there's two different ways the main chain can interact. What else is important? Yeah, so the interactions that we're looking at still are main chain, and in this case, we're looking at hydrogen bonding.
Yes.
Yeah, hydrogen bonding.
I think they draw the carboxylic acid proteinated.
You see what?
The carboxylic acid might be proteinated. Who knows? I don't too want to ask about that. It doesn't matter. Because the way you did it would technically be ionic.
Yeah. All right. So these right here, these bonds between these amino acids that you see right here, the dotted, the dashed lines, that represents hydrogen bonding between the main chains because of the attraction that an O group has to a hydrogen group. And yeah, that's mostly what you're gonna be tested on for secondary. And so now we'll move on to tertiary. But if you guys have any questions about this at all, post it in the comments.
Yeah, I would say the important thing for these is just to be able to identify it in any particular situation.
And the interactions.
Yeah, so she might throw like a long, convoluted problem at you and just simply ask, what protein structure is this?
Yeah.
So how do you distinguish between the two? I focus on those things. I hope my voice gets picked up.
What's up?
I hope my voice gets picked up.
Tertiary is when we have, let's say, how do they usually draw? Is it a bond? Or no?
For ionic?
Yeah. Or they just draw it close to each other, right?
Yeah. Parentheses.
Um,
Say something.
and then so when we're looking at when we're looking at tertiary bonds, those are the ones. Oh no, some glare. Is that better? No, that's not better.
You do it like right on the
Yeah. Dang it. Take up the glare. Can you take off the glare?
It's in line.
Nah, it's hard to take off the glare. Okay. But anyways, so if you guys can see it, I'll put it closer. Maybe that'll help. All right. So this one right here, we have a bunch of hydrophilic groups that are grouped together that are attracted to the other ones that are hydrophilic, and they're attracted to the polar environment. So you can have hydrophilic interactions. And now notice that I drew bonds coming out of these amino acids. That's because when we're looking at the tertiary level, we're looking at the R groups. That's when the R groups start to come out and start to shine because those are the ones interacting now at the tertiary level. So we also have these over here, these very hydrophobic molecules kind of group up, and that's what we call hydrophobic interactions, right? They're interacting with each other and trying to group up close to each other. That way they don't have to deal with this polar environment that they're in. While hydrophilic interactions are occurring like here because these want to interact with things that are hydrophilic as well, and also with their environment. Lastly, next we have this right here, where we have a positively charged and a negatively charged atom. Not atom, what am I saying? Molecule. And these are attracted to each other because of the positive and negative charges. And so what you'll see between these is ionic bonds. But next we have, when two amino acids have sulfides or sulfur groups next to each other, close enough where they can create a bond, you'll see a disulfide bond. So in this, we have two S's joined together, and this is the only type of covalent bond you're going to see between R groups, which is important because I'm sure you'll have a question about what happens when you denature things, and the only thing that stays alive the only thing that stays intact when you denature things are polypeptide bonds or peptide bonds, because those are covalent bonds, and this disulfide bond because this is a covalent bond. Next, we also have hydrogen bonding. That's going to occur because partial positives, partial negatives are going to be attracted to each other, and I believe that's about it. That is tertiary interactions. That's tertiary. What else?
You need questionnaire?
Hmm?
You need questionnaire? Also, you got any question, but I made a list.
Okay. Cool. Yeah, so that's tertiary. And so basically, so far we've covered primary, secondary, tertiary, and now we're going to go to quaternary. But in each one, we're kind of increasing the complexity of things. So in the first one, we only had peptide bonds. And so we had these bonds between amino acids. And now once we had a protein, now it's starting to curve, or it's starting to form a shape, because some of the proteins or some of the amino acids in that protein are attracted to the other ones because of hydrogen bonding, and that's where you create alpha helices and beta pleated sheets. And then for this one, now we're getting even more complicated because now we have R groups interacting with each other from the main chain, and we have all these types of interactions. And so now we're going to move on to quaternary, and that's when we move on to just having looking at one protein, to looking at two. So let's say we have this protein here, and then
You might have to switch to the green.
Really?
Yeah, it's like okay.
Okay.
But I feel like the next thing.
Yeah. So when we're talking about quaternary, or at the four degree stage, this is just an abbreviation for quaternary. But when we're talking about quaternary, that's when we get even more complicated, because we have one protein that has the peptide bonds. From the primary, we have the same protein have hydrogen bonding between the main chain, and then we have the same protein doing R groups or having R groups interact with each other. And we have all that complexity, and now we're going to introduce that to another protein that has all those three things as well. And so what we see is that in the quaternary stage, we can have two proteins interact with each other, either through disulfide bonds, ionic bonds, or hydrogen bonds. And we can also have these proteins interact with much more. So we can have polypeptides that are interact with like three other molecules or three other proteins, and they all interact to create one super protein. That's probably what you're going to see in hemoglobin. Is that correct? Yeah, hemoglobin, which is a protein that you might have heard of. It has to do with your blood and all that stuff. And that one's made out of 4 subunits of proteins.
Tyrosine kinase receptors.
Oh yeah, tyrosine kinase receptors are also an example because those are made out of 2 proteins before they dimerize, right? Is that correct? Yeah. Okay. What else?
I think she's asked that before.
She's what?
I think she's asked that like a dimerized tyrosine kinase receptor is what level protein structure?
Okay.
That's like a way she can like reach units.
Yeah. Okay. Yeah. So what Derek said was that the tyrosine kinase receptor. Remember how it starts off as 1? Starts off as 1, right? And then as soon as it gets its ligands, it dimerizes, right? So that's an example of or when it dimerizes, that's an example of a quaternary protein, quaternary level protein, because we have 2 proteins that are interacting with each other. But besides that, that's about it. And this one, those are the ones you should be worried about. So hopefully that answered your question, Yesenia. Let me know if you need further clarification. But we're going to go on to photosynthesis. Let me erase this.
Okay. And Jerry has a question too, but I got to pulled up for you. For the cloning stuff.
Oh, that's cloning. Okay.
I'll leave it.
Yeah. Could you just give me 1 sec? I need to pull something up.
Yeah.
Yeah, but I gotta look at photosynthesis.
Oh yeah, you're right.
Chapter 9. So when we're talking about photosynthesis, we can talk about it in terms of what's occurring where. We can start there. So in the beginning, oh, and it's green for photosynthesis.
Nice.
Did you even plug that? What was the other one? Can you see that there?
It's okay. Let me try turning the lights.
All right. Turning off or turning on the lights? Can you guys see that? Let me know in the comments. Oh, that's better. I guess, yeah. Is that better, guys? Like, can anybody give me like a quick thumbs up or quick down? I think that's better. Oh, is that better? It's too light of, yeah. All right. The black one's running out, but let's see how much better this is. Yeah, I think the black one's still better.
Okay.
Yeah. This is a good setup, though. I almost feel like we need a fireplace or something. This is nice.
Yeah. I'm gonna lower the blinds.
All right. Let's see. Oh, H. Is that too? I think it might be a little bit too dark. Yeah, I think it was better with the shades up a little bit. Yeah. um, so yeah, so when we're talking about photosynthesis, we can start off by talking about what's going on and where. Oh, okay. So when we're talking about photosynthesis, we talk about the whole thing occurring in this thing called the chloroplast. Similar to how respiration, when we're talking about respiration, we're talking about it occurring in the mitochondria. The plant, the plant's photosynthesis occurs in its similar kind of organelle called the chloroplast, where the chloroplast is this giant bean-like thing, kind of like the bean that the mitochondria looks like. Chloroplast. Well, this is the thylakoid. Why are these? All right. That's the thylakoid that we look at. This is the thylakoid, and then this is the chloroplast. This whole bean structure is the chloroplast. What happens in the thylakoid is that we introduce it with some H2O, and then we get out O2, and we'll go into the process. We'll go into detail of the light reactions that occur here, and then after that occurs, we have some interactions between the light reactions and the Calvin cycle. Light reactions in the Calvin cycle because they interact through the use of NADPH molecules. Similar to how, if you think about it, in glycolysis and pyruvate, similar to the TCA cycle in mitochondria, they create all these NADHs and all that stuff, and then they transport those NADHs to the electron transport chain. That's kind of what we were going to see going on in the light reactions and also in the Calvin cycle. Yeah. So there's a lot of correlations between the two different processes. So now we can go into the details of the light reactions. So let me erase this real quick. Oh, the last thing was, what do you need to know? Stroma. That's so crowded. Erase this.
You draw a bigger bead.
Yeah. So the last thing we'll talk about is I just wanted to mention the anatomy of this. So basically, when we're talking about this, we're going to talk about, is that legible? Yeah, this disc right here is a thylakoid. Everything inside the chloroplast, there's like a kind of a liquidy substance inside the chloroplast, and that's called the stroma. When you have a stack of these thylakoids, that's called a granule. Yeah. Okay. And then the whole thing is the chloroplast, and then so the reason why it's important is because when you're talking about light reactions, and when you're talking about this, where each thing occurs, you're going to probably be asked like, oh, where is the Calvin cycle occurring? So you guys can see that it's not occurring in the thylakoid, it's occurring in the stroma for the Calvin cycle. And then you'll see it's also going to be important when you're talking about the electron transport chain, because of the changes in pH in one region versus the other. And yeah, but that's basically all the terminology you need to know about plant cells. And so let's move, or chloroplasts. So now let's move on to the actual light reactions. Oh, just give me one sec. All right. So much better, right? All right. So
Basically, but it's like cleavage. So you just insert it into the bacterial. You put a plasmid. Somehow use the plasmid to. So you cut out restriction enzymes. Interesting that. I don't know if it's talking about inducible. The whole like.
what else?
I'm not sure. It's more. Dude, David, that's so much better.
So much, right?
Also, watch
You
out
can-
here.
Huh?
Don't make it. Yeah.
All right. So let's see. So this is.
Six is a wavelength, right?
Yeah, it refers to the wavelength of the photon. What else am I missing? Derek's going to lead the explanation of this. The only thing is that I'd mention is that when we're talking about pigment or P700 is that this P stands for pigment molecule. That's what this is. And then the 700 just refers to the wavelength needed for that molecule to be excited. Take it away. Actually, let me get my Yeti cup.
Seating seals. Oh, sick, you can't. Okay. All right, so we know, okay, we'll start over here. So we know that light is basically going to start a reaction, and it's going to be picked up. So there's a diagram in your book. It's like green and purple or whatever. And so light is going to come in and like bounce across these like photoreceptor pigments. But ultimately, it's going to excite this P680 right here that David has drawn. It's like the two molecules. And so when it does that, it excites P680. An electron it's going to go from P680 to its excited state. So think back to Gen Chem, like when you receive energy in an electron, it goes to an excited state. So the thing is with P680 is okay, so I don't know if you can see that, but it's P680 to P680 star. The star just signifies that it's excited. So when it's excited, it's not very stable. So it's going to give up an electron because of all the additional energy it's acquired. And so when it does that, it's going to give up its electrons in the form of a hydride to PQ here. And there are better diagrams like in your book that I highly suggest looking at. So it's going to give it to PQ, and once it gives it to PQ, it becomes PQH2. And then, do you have that picture? What did that say? Oh, just kidding. Okay. It's going to give the electrons to PQ, and then PQ will give it to cytochrome. And this is all within the membrane. So once the energy, just like in the electron transport chain of respiration, once the cytochrome receives the electrons, it's going to use that energy to pump out another hydrogen. So if we look at, our markers are really bad, but this whole complex is within a membrane. This would be the outside, this would be the inside. And so just recap, light hits it, excites electrons, gets to P680, P680 becomes excited, and when it becomes excited, it will deliver its electrons to PQ. PQ will deliver to cytochrome C, and that will use that energy to pump out hydrogen. And then after cytochrome, it'll go to PC, which will then deliver its electrons over to P700. So, okay, so if you think about P680 for a second, right? So we know that it gets excited, and it once it gets excited, it delivers an electron to the next molecule in the chain, right? But after you deliver an electron, P680 is now missing an electron. So hopefully that makes sense. So like you give something away, now you no longer don't have that, right? So it got energy, it gave an electron away, so it's a deficit of one electron. Well, the problem is if we want to continue to do this electron transport change, we need to replenish that source of electrons. So that's where water comes in. So P680 that's missing an electron, we call that P680 plus. You don't have to know this for your exam, but for me, it's just easier to differentiate. So when P680 has one less electron than it's supposed to, it really wants that electron back so much that it's strong enough to pull an electron straight from water to become the normal P680 again. So when you're down an electron, P680 really wants it back. It'll rip one from water and become the normal P680. So once sunlight hits it again, it can become excited, go to P680 star, and then deliver it again. So that's how that process repeats and repeats and repeats. And same thing with P700. You also have other photoreceptors here, and P700 will get excited and deliver its electrons to ferredoxin, correct? P700 ferredoxin.
Yeah, ferredoxin.
Ferredoxin, FZ. So same scenario there. It gets excited, and once it gets excited, it's no longer as stable, so it will deliver its electrons to the next thing in the line. But when you do that, okay, you preceded the reaction, but you're now at a deficit of one electron, so we need to get that back. And so in order to get that electron back, we have PC from this original reaction, because remember, we did deliver the electrons. They're still coming, but we kind of stopped at PC. So PC will deliver it back to P700, and to reset it as the normal P700. And so therefore, like once the electrons hit it again, the reaction can repeat itself. Because if you can only do it once, then there's no point. So that's why we need to keep on refilling the process. Let me know if that makes sense in the comments. I believe it's like over here or something. Yeah, so if you guys want me to explain that again, I definitely can. And then we can erase some stuff and then talk about some cyclic versus non-cyclic flow. Okay, so basically after that whole we're like transferring the electrons, after P700 gets excited, it's going to deliver its electrons. I don't know if you can see that well, but to FD right here, it's kind of a blur, but ferredoxin, and then ferredoxin will give those electrons to NADP plus to produce NADPH, just like we saw in respiration. And that's how we make NADPH. And then if you remember back towards between the two photosystems, we have, once we transferred that electron to cytochrome, we're going to use that energy to pump out a hydrogen. So now like as time progresses, we'll have a bunch of, try to stop looking. Oh yeah. We'll have a bunch of hydrogens on this side of the membrane. And so just like in the electron transfer of respiration, we have a bunch of those protons. We can also have, I guess, so if you remember, we're going to pump a lot of hydrogens on this side. So a lot more on the inside of the cell. This is the inside, this is the outside. And naturally what they want to do is flow back out to reset the gradient, because remember, nature hates gradients. So what these guys will do is they will all begin to flow back through ATP synthase. And if you recall, ATP synthase will use, it's hollow, and so once hydrogens flow in there, it spins. It's like those things that, like, you know, when you go to like Six Flags or like the subway and you walk through and it like spins and like counts you. That's exactly what ATP synthase does. It's also like a water wheel. So like as things flow through it, it starts spinning, and it uses that energy as it's spinning to jam an ADP and a phosphate together to make ATP. And so, yeah, we're going to create a bunch of ATP because of the light powered reactions in order to pump protons across the membrane to go back and flow through, to use chemiosmosis once again. If you recall, like there were like two ways to make ATP in respiration. It was substrate level and oxidative phosphorylation. This is the third method. This is called what is it? Photophosphorylation. It's photosynthesis. Yes, the method to make ATP. Yeah, photophosphorylation is basically light powered, and hopefully you can see how that makes sense. And now, so we were talking about cyclic flow. This is non-cyclic flow, by the way. So that makes sense because everything that we talked about in this, there's no cycle. It's just like a progression that keeps on happening over and over. But in cyclic flow, what we're going to have is, you guys can see in orange, ferredoxin, instead of giving its electrons to NADP plus to make NADH, what it's going to do is going to deliver its electrons all the way back to cytochrome. And so what that does is once we deliver electrons back there, it's going to use that energy to pump more hydrogens back into the space, the inside of the cell right here. And in doing so, we're going to have more protons down here. And then we create more of a gradient, so more protons can flow back through ATP synthase, and we can increase production of ATP. So hopefully you can see that once we do that, if ferredoxin no longer gives its electrons to NADP+, we're not going to be making NADPH, but instead we're going to be using it to like mass pump H plus ions outside or through the membrane, excuse me. And then so we can mass produce ATP. So hopefully that makes sense. You can possibly try to think of like some scenarios in which we would do cyclic versus non-cyclic flow. For example, if we have a lot of NADPH, do we think you'd want to do cyclic flow or non-cyclic? Okay, but hopefully if that doesn't make hopefully if that doesn't make sense, hopefully it does make sense. And if it doesn't, you can let me know in the comments and we can go over it again.
All right. So now that we went over the photosystems, we can go through the Calvin cycle, and then that'll be the last thing to talk about for photosynthesis. And then we'll move on to DNA cloning, because that was the next question that was asked. Give me a second to finish drawing this. All right, so we're just gonna quickly go over Calvin cycle. We're not gonna go into depth about each single molecule that is a part of this cycle because you're not gonna get that tested that deeply on this. So it's mostly important. The things that are most important is what's going on generally through the cycle, the phases, the the products, the reactants, and stuff like that. So we'll go through that. But don't worry, if you want to get tested on that, you'll get tested that on biochemistry. So you'll get your chance to get tested on it. So for the first part, when it comes to the Calvin cycle, if you guys remember when we drew it, generally we had when we drew it generally in the first half or the before we even started talking about photosynthesis, we talked about the Calvin cycle being in the stroma, and we kind of drew it as this general CO2 coming in, sugar coming out, and so that's what we're going to be focusing on right now. And so when we're looking at the Calvin cycle, I'm here. When we're looking at the Calvin cycle, the biggest thing to remember is that we're taking in CO2 molecules, which are represented as this black dot, I guess, or black ball. Yeah. So each one of these represents a carbon, and in this case, this represents a CO2 because CO2 has a carbon. So we're going to accept 3 of these, but they're going to be accepted one at a time, which makes sense because for each one of these, you need a carbon, a CO2 acceptor to accept it. And so that's where this RUBP is. RUBP is just ribulose bisphosphate. Ribulose bisphosphate, so ribulose bisphosphate, BP. And so that molecule accepts CO2. The enzyme that catalyzes this reaction, this accepting of CO2, is called Rubisco. Those are 2 things that you have to know, that you have to memorize and know for the exam. Definitely know that. What happens is carbon, and once it's accepted, it goes through through the Calvin cycle, and the Calvin cycle is broken down into 3 phases. The first phase we call carbon fixation, and so the way you can kind of think of it is that carbon, it's going from CO2 and it's being fixed. Fixed with what? Well, most of the time, when we talk about fixing molecules, we talk about adding phosphate groups, and so that's what we see. We see ATP being converted to ADP, where the phosphate group is added to the molecule. What's it called? I forgot what that molecule is called.
CO2. We're fixing it.
Am I including? Oh, okay. The ATP step is in reduction, right? No, wait, no, that doesn't make any sense. Yeah.
Okay. Okay.
Yeah. So carbon fixation is both the carbon being accepted by the ribulose bisphosphate and also ATP adding a phosphate group to that molecule. Now, the next step, the next phase that it goes through is called reduction. And so reduction, basically the way we've seen reduction is through these electron acceptors. And in this case, it's no different. We're looking at, you can't really see that. We're looking at NADPH, which is similar to NADH, which you see in animal cells. The only thing is this is the plant version of it. And so NADPH, like I mentioned in class, you can remember this is the plant one because it has a P in it, for plants. And so in this phase, in the second phase, we go through reduction. But because we're talking about the carbon, this NADPH molecule isn't going to be reduced. It's going to be oxidized. So this is oxidized to NAD. You can't see that. NAD plus. Is that better? Yeah, you can see that a little bit better. Yeah. So the NADPH is oxidized to NADP plus, While that's oxidized, the molecule that's going through this Calvin cycle is going to be reduced. And so that's why phase 2 is called reduction. Now, after all that occurs, we're able to create this one of these molecules. There you go. One of these molecules that's called G3P. And so basically that means that there's 3 carbon groups, 3 of these dots, attached to a phosphate. And that's where the name comes from, G3P. G3P serves as a precursor. Thank you. As a precursor to glucose, because as you remember, the formula for photosynthesis is basically the opposite of cellular respiration, in which you create glucose in photosynthesis, while in cellular respiration you use glucose. And so this will serve as a precursor to create glucose. Now the thing is, once you go through reduction, you're going to create a total of 6 of these G3P molecules, but because you want this to be a cycle, you need to recycle things. So only one of those 6 is used as a product, and the other 5 are brought into the regeneration phase. And so in the regeneration phase, those are phosphorylated again, and they're phosphorylated again, and they're creating. In the regeneration phase, you're creating RUBP. And you're in the regeneration phase, what it's referring to is the regeneration of RUBP, or ribulose bisphosphate, because you need this molecule to accept the next round of CO2 to continue the cycle. And so that's about it when it comes to the Calvin cycle. You don't need to know every single molecule, just really understand that the enzyme, remember ribulose bisphosphate, remember the CO2 comes in. There's three of them are needed to go through the cycle, but they are accepted one at a time. Know the phases, know what occurs in each phase, and then, I know the reactants. So the reactants in this case would be 9 ATP molecules for 3 CO2s and 1 product of G3P. And then 6 reactants of NADPH for the reduction phase. Should I draw that out as a formula? Yeah, okay. But yeah, so just remember the products, the reactants, what's going on in each, what each phase is and what's going on in each phase. And that should be it. That's all you should know for photosynthesis, in addition to what Derek said about the photosystems and the cyclic flow and non-cyclic flows. But now we're going to move on to DNA cloning, because that was the next question in the series of questions. And David's going to take over for that. But yeah, if you have any more questions, put it in the comments, and we'll get to them. Yeah. Oh, let me erase this for you.
Good evening, everyone. How are you?
All is well.
That's good. That's good. Thank you. Thank you, Dave.
All right.
So right now we're going to be talking about cloning. So when it comes to cloning, we think about DNA, cloning our DNA. So in terms of the cloning process for gene cloning, we want to focus on 2 DNAs. We have our DNA from our bacteria. We'd call it BAC. And we got our DNA. DNA for our gene of interest. So let's call this one enter. So our bacterial DNA we know is a plasmid. Yes, it is a plasmid. And then we have our DNA of interest. So what we're gonna do is, for the gene process, so specifically this is for you and Jetty, that we're gonna extract our bacterial DNA, right? And then we're gonna look at our gene of interest from our organism and extract that particular gene. So in this case, it'd be this. And what we're gonna do is we're gonna open up plasmid.
It's hard to see your orange. So draw a little lower because you're the clear.
Gene of interest. And what we're gonna do is we're gonna then insert that gene of interest into our plasmid. And yes, for example, and one thing to clarify too. So if this gene of interest is from another bacteria, in this case, we wouldn't need to go through any processing. We can directly splice it out and put it in. But for most cases, we're going to be dealing with genes from other species, like from eukaryotes. So in this case, can you directly insert a gene from a eukaryote into a prokaryote? No, you can't do it. So what we need to do is go through a type of synthetic RNA splicing. In this case, just genetic engineering to alter the gene from the eukaryote to make it such that it can actually make a functional protein when created in the prokaryote. So that's what's going to be happening here. So let's do that. So we got genetic engineering. To make a functional gene. A functional gene is then inserted into the plasmid. That plasmid is then reinserted into bacteria.
Are those like the sticky ends that we're talking about?
Oh yeah, I was going to clarify that.
Okay. It's like, when you talking about genetic engineering, I was like, oh yeah.
It's going to be inserted into there, and then we know that bacteria have an amazing ability to replicate. So as it creates the proteins through like the bacterial life cycle, when it's translates the proteins from the DNA, transcribes and translates the proteins, it's going to create a whole bunch of proteins, and those bacteria are going to then multiply, create more proteins, create more proteins. So now with our gene of interest, from our interested organism, we inserted that into bacterial DNA, and then we have bacteria which do the work of creating that protein in mass such that we can use it in other experiments. That makes sense? Njede, are you good? Are you good? Are you still here? I hope that answers your questions. So next, we're going to be moving on to macromolecules. So Derek, can you tell them what's up?
Oh yeah. Oh, sweet. He said yes. All right, good evening. Hey, David, which macromolecule do you want? Not proteins, though, because we talked about that in the earlier times of this video. So
it's all good.
Yeah.
I'm feeling something sweet right now.
Oh,
so
so lipid. I'll play it. Okay. Yeah, so we'll talk about carbs. So basically, carbohydrates are just. Oh wait, Andrea has a question. Does the gene of interest have to be from a eukaryotic organism?
So to clarify, Njede just asked, does the gene of interest have to be from a eukaryotic organism? No, it does not. In this case, if it is from a eukaryotic organism, then we need to do what I said before, the genetic engineering. But if it's from another prokaryotic organism, we wouldn't have to do the genetic engineering.
Yeah, and just to follow up on that, all DNA is the same. You got your A's, G's, C's, and T's across the board of all organisms. That's the same, so you won't have to do much. The reason why we do genetic engineering for eukaryotes, because think about the DNA of eukaryotes versus prokaryotes, right? As you recall, we'll have introns in our eukaryotic DNA. But if we want to express that in bacteria, well, that won't work as well because bacteria don't have any introns. So you need to do things to remove said introns, or get a piece of, or find a way to get a DNA that doesn't have that in general, and to put it back into the bacteria. So because those introns, when bacteria try to express it, it will affect with the protein. So hopefully that makes sense too, and that's a little nod to exam 2 material. But we will continue with carbs. Oh, you like that?
Mm-hmm.
Yeah, right? Pay no attention to us. Okay. Yeah, so basically carbs are just they're cyclic ring molecules with usually 5 or 6 membered rings with an oxygen in the corner. And And then if you recall, if we count our carbons, we'll have 1, 2, 3. In this, oh, you cannot see that. I apologize. Are there problems with these carbons? I don't think so. Yeah, but if you can count the carbons, we've got 1, 2, 3, 4. That's a 5 and 6. And same thing for here. 1, 2, 5. It's 5-membered ring. Okay, and so basically with carbs, because I mean they're just sugar molecules, right? We just break them down for energy, as we saw back in the respiration stuff. What you want to focus on for carbs is how the structures are different, and how will that overall affect like the polymer structure or Pauli range. So for example, we'll look at glucose because that's the main carb she focuses on. Okay, so we see this. So, for example, if you saw this on an exam and she asked you to label this, what would you label that as? Most people would typically just put glucose, right? But there's more you can do it to that. So, did we just learn about one type of glucose in this class? No, we didn't. We learned about 2, alpha and beta. So if we look at carbon number 1, we see that our OH is pointing down. And if you recall back in the day, we used to say batter up. So if the glucose is pointing up, it's a batter. And so batter starts with a B. Yeah, batter's holding bats up, batter up. And the B for batter is for beta. But in this case, it's down. So when we talk about up or down, like let's say if you want to cut this in half, the virtual plane right there. If it's below, that's down. If it's up, above, that's up. You can see that this is clearly below that line. And we know that this is an alpha glucose. And you can't really tell. It's kind of annoying, but this is just like a poorly drawn 2D image of glucose. But in reality, these molecules are in 3 dimensions. So the whole up or down orientation really does make a big difference in this case. It makes a bigger difference when we talk about how 2 glucose molecules are interconnected. So,
You need to remember
what?
the numbering of glucose.
Should they remember? Okay, so for this purpose, I didn't draw the other OHs just because there's really no need. But who can tell me what this bond is right there between these 2 carbohydrates? And I'll wait. I'll wait for it. I got nothing to do. My finals on Saturday. I'm good. Yeah.
Does it count if I put the answer?
Oh, you know what? Maybe I'll allow it. My laptop's at 31%, guys. You better hurry. Glycosidic linkage. Yes, that is correct. So, but not so fast. Just like with before, there's an even more specific way you can answer this. The reason why I emphasize this so much because when I took this class back in fall of 2015, she had a question like this on exam, and she said, what is the name of this bond? Basically, it's the exact same question. And I put glycosidic linkage. That was wrong. It was partially right. But the real answer would be what we think is. Is there a way we can further classify this? Well, if you know earlier, we said alpha was down and beta was up. And this also applies to glycosidic linkage. So if we have the linkage, so we have our sugar here and sugar here. You see how it's pointing down? There's an arrow going down. Well, that would be an alpha. And the same thing. I realize now these are all just circles. That'd be beta. And just like we said earlier, batter up. So we're pointing at the little triangle. Hopefully you can see the triangle right here. Now it's going down. And then up here it's going up. That's beta. So the real answer to this question, the real answer to this question would be an alpha glycosidic linkage, because we can further classify that. And so this is important because there are two types of glucose polymers that have alpha and beta linkages, and it can be a sec to draw it. But just because they have that one difference, up versus down, it forms a completely different structure. All right. So if you recall, if you're looking back at this in like a week from now, take a second, pause, and try to figure out which polymer of glucose this would be. Which one is cellulose and which one is glycogen? Yes, Injury, that's perfect. That's exactly what we're talking about. Oh, also, it's a 1,4 alpha glycosidic linkage because, real quick, side note, yeah, so Injury, this is a good point. If you label the numbers in your carbon, one side of the bond will be number 1, and the other side will be number 4. So 1, 4, 1, 4 linkage, right? And then we also have. Okay, so we can see that there's a 4 and there's a 1. So this right here, that's our alpha 1,4. But we also have another linkage that determines branching, and that one would be the 1,6. So 6. So you can see on this carbon, we have, hopefully you guys can see that, on the 6th numbered carbon of this carbohydrate, in the 1 on this one, we have another one connected. So if we have a bunch of 1,4s together, they look like this, right? So if I had a bunch of these together, we just line it up in a linear chain, right? But if we had a 1,6, well, if you note that the 6th carbon is pointing up from here, it's like kind of like an antenna from the ring. So if we have a bond, something bonded to it, so you can imagine that each of these guys have like an antenna. If we have a 1,6 linkage, there's another carbon coming off from here, and we could have other things from there. But for these purposes, we'll just. And then from that 1,6 linkage, we could have other 1,4 linkages on that too. And then there's another one. So hopefully that makes sense. So each bond between the circles right here, that's a 1,4. But the branching points right here and right here, those would be 1,6. And they create like another like diverging pathway. It's like a fork in the road. Hopefully that makes sense. So that 1,6 linkage is really good for branching. And then now we'll go back to so if you're gonna look at this, so the answer to the question earlier is this structure would be cellulose. And this would be glycogen. So if you're reviewing later down the line in the week, take a second and try to figure out what kind of linkages are in, or glycosidic linkages are in glycogen and in cellulose. And there can be multiple ones for each molecule.
Branching is alpha.
All right. So glycogen will have alpha 1,4 and alpha 1,6. So if you're going to zoom in on this guy, so let's say we zoom in on this plane, you can see that those will just end up being a bunch of circles together. Hopefully you guys can see that. And just like in that last example I drew earlier, the bonds between those little circles would be the 1,4s. But if you look at an area, let's say right here, you're going to notice that we, in addition to having circles connected this way, we're also going to have another branch point, which is this long line right here. And that's the 1,6. So as you all know, glycogen is mainly used for energy storage, right? And so have you all ever played that, it's that game where it's like a bunch of like balls coming down and you're at the bottom and you're like shooting colored balls to like match the top and then like stuff falls down. If you ever hit like, there's like a branch point, and there's like, what I'm talking about, where it's like, gosh, look at me like I'm stupid. But like, you're like shooting balls like up this way, and you're trying to like hit combos or whatever. And you ever done that thing? Have you ever done that thing where like, if you've hit like, there's like a little part left that's like makes like a large part hang, and you hit that with the colored balls, like the whole thing falls, and you get more points or whatever? That's basically glycogen. So if I need a fast storage of glucose to dump into my bloodstream, it's definitely not effective to go and to zoom into every single spot and begin to break like the bonds between each circle. I would just start breaking it, let's say like right here. And now this whole branch piece would just fall off, and I can begin to break down that in other places. So in other words, glycogen is very branched because there's multiple access points for enzymes to come and break down these linkages in order to like use the glucose, because you're going to break them down to individual pieces so you can use it in the blood or transport it in the blood to your various mitochondria in your cells and whatnot. Over here in cellulose, notice that we don't have any branching going on. We just have the beta 1,4. And this is kind of difficult to wrap your head around because once again, we're looking at 3D structures from a 2D source. And you'll see more of this in OChem, but the way the beta 1,4 linkages will line up in 3D space is that there will be hydrogen bonds between each of these little chains. And so you'll have like nice linear stacks of sugar molecules that are hydrogen bonded together like this. And so think about like what kind of function that would serve. And it'd be like support beams for plants, because think about plants don't move, then you have strong supports. And that's basically what they do. They have long glucose chains, and they'll create these long support structures in cellulose. Excuse me. But glycogen, on the other hand, it's very branched, so any enzyme could break apart anywhere, and now you can deposit, or you can begin to break down this branch of glucose. Because remember, these are the same molecules except for that one bond of the 3 carbon. It's just slightly different. But hopefully by now you guys can see that just by changing 1 carbon on a glucose molecule, you can have very different structures with very different functions. And also, I guess, important to note or remember, can we break down cellulose as humans? The answer is no, because we don't have an enzyme that breaks down the beta glycosidic linkage. All right, so if that makes sense or doesn't, please let me know. But if not, one of the Davids, never guess which one, we'll talk about another macromolecule. Yeah. You're muted, David.
I'm muted. Oh,
You're
okay.
good.
Hi, I'm back. Now we're going over. Nucleic acids. So I'm not going to start this out. You guys are going to start this out. So can you guys tell me the structure of a nucleic acid? Roman, you are correct, my dude. So we are starting out. Yes, we have our phosphate. Then we have our sugar. Sugar. And then we have our nitrogen space. In this case, can someone identify to me which specific type of nitrogenous base this is, purine or pyrimidine?
I don't think they would know that.
Well, in this case, it is a pyrimidine. We will go over mnemonics for that later. But in this case,
Purine.
purine.
Little name, big structure. Big name, little structure.
Yeah, this
Purine
is a
versus
purine because this is the base. It's got 2 rings. If it was just 1 ring, then that's a pyrimidine.
Good way to remember that is that pyrimidine and purine, one word's bigger, one word smaller. So pyrimidine goes with the bigger structure, which means 2 ring.
Oh, we're on.
Pyrimidine
Pyrimidine is
goes.
a bigger word, smaller
Little
structure. Purine is smaller word, bigger structure.
I apologize, but that's why teamwork makes the dream work. Thank you. In this case, so we know nucleic acids serve two major functions: is to store genetic information and transfer genetic information. In this case, the one last thing to go over for this is that exactly what Roman was saying. That's that we have a phosphodiester bond that happens with the pentose sugar with the adjacent phosphate from the from like our adjacent nucleic acid. So in this case, bonding.
Yeah, there's a.
In this case. So for this test, as just a refresher, we're not going to be going into as much detail as we did for the first test. So this is basically all this is just the main stuff you really need to focus on. So anything else you want to add there?
Yeah, so you guys, I don't think you know this yet, but an ester is basically, you have like, it's typically carbon, and then this is like super throwback to exam 1. You have a carbon double bonded to oxygen, and then you have single bond to another oxygen, and then we say R. R is like, think about like X in algebra, how X can mean any number. R is like the OChem version of any molecule possible. So if you see that, we have a really similar here. And to break down the word phosphodiester. So diester. So if this is a single ester, if we had to make it a double, we would just go this way. So if you look on just the left and right side, you'd see that carbon double bond to oxygen to another oxygen in our group. That's one ester. If you You go on this side, it's the exact same thing. Well, but it's phospho. Well, remember I said there's a carbon here, but we can also replace that with a phosphorus. And that makes it a phosphodiester. And we can see that here. We have a phosphate double bond and an oxygen. Actually, we have to do that as well. Double bonded to an oxygen, both sides, and then we have single bond to oxygen, and then this right here would be our R group, and then that right there would also be our R group. So hopefully that name makes sense. If we had to draw another one on this side, oh god, yeah, it'd be like God, I can't draw. Yes, you guys can't see that. But yeah, you see how it's. Oh, also important to note. And hopefully this will make more sense. If we had to number this ring, just like we did for our other things, we'd start here. See. Oh, actually. We messed up. That bond goes to that carbon. Sorry about that, guys. We caught our mistake though. If you number the carbons, you remember in exam 2, it's always 5' to 3'. That's where the phosphate will bind to in the ribose sugar. So we have 1, 2, it's on the 3', and the 5'. Those are the two ends that will touch the phosphates. And if you think about if you have these in a chain, so let's say these are interchanged. So up here. So if we end this chain right here, like one end will always have the 5'. It's like the head, and then at the very bottom we'll have just the 3' ending exposed. And that's like the tail. Really similar to the N and C terminus. You know how like no matter how long your polypeptide is, you're always going to have the first one is the N terminus and the last one is the C terminus. That's the exact same thing with here. We have the 5' exposed here, and then the 3' somewhere along at the end. And that's how you can read the direction of DNA. And that's the whole. We read 5 to 3. Synthesize 3 to 5, or sorry, read 3 to 5, make new DNA 5 to 3. That's the orientation we're talking about. So from there to there, or there to there. That's basically it for nucleic acids. All right. So if you guys have any other clarifying questions, please feel free to ask. I know we actually modeled the second carbon, but hopefully you guys can see it's on the third one. If not, then I'll leave it over to David to finish up with lipids.
So, all right. So the last thing we're gonna talk about today is lipids. And so to talk about lipids, we can talk about it in the sense that lipids is the last category of macromolecules. Uh, Even though we classify it as a macromolecule, it's very different than proteins, carbohydrates, and nucleic acids because even though it's a macromolecule, you can't see that, but it's not a polymer. And what that means is that it's not made out of one individual subunit that's always the same. And you'll see why once we get into glycerol and all the other, or phospholipids and all the other combinations of that. But the monomer in this case, even though it's not, or the most simplest building block of lipids, wouldn't be classified as a monomer. But what we know is that the building block would be a fatty acid, and for this one, the way I drew it is it has a carboxylic acid group, and then it has a carbon chain. It's in this carbon chain, even though it looks like a zigzag, it's just an abbreviation of carbon to carbon to carbon bonds with hydrogen groups. That's how we abbreviate this fatty acid. So in this case, when we have this right here, this is what we call a saturated fatty acid. The reason why is because when we're talking about saturated, think of like drenched in or like having so much of it. In this case, it's just saturated with hydrogens. For each carbon, there's two. But now, on the opposite sense, when we say unsaturated, we can mean that. Well, sorry. What that means is that it doesn't have the maximum amount of carbon or of hydrogens that it's possible, because for this one, each carbon has two hydrogens. But for this, some of these carbons, specifically the ones that are involved in the double bond, are not going to have two hydrogens. They're going to have at most one. So for this one right here, if we look at this carbon right here involved in the double bond, it goes from having two hydrogen groups attached to one carbon to having having just one hydrogen group attached to one carbon and one hydrogen group attached to one carbon because of that double bond. And because we know that carbon will only have 4 bonds, and if 2 of those are attached to the other carbon, then there leaves 1 for hydrogen and then 1 for the other carbon that it's attached to in the chain. Last thing is, though, or the next thing is unsaturated FA as an abbreviation. The thing when it comes to unsaturated, it could be at least 1 double bond, or it could be 4 double bonds, or it could be 40 double bonds, as long as there's enough carbons. And so that's why that amount of variability, that's why we're not allowed to say that lipids are polymers. We could say they're macromolecules because once they form together and they're created into like the phospholipids and all that stuff, they're very, very big molecules. But in the individual level, there's so much variability for unsaturated fats. Like it can have 1 double bond, it can have 40 double bonds, it can have 30 double bonds. It has a lot of variety, and so that's why we can't single it down to a single monomer like we do amino acids. Even though they have the R group, they have almost the rest of the molecule the same. And so that's why we're allowed to use, or that's why we're allowed to say proteins are polymers of amino acids, but we're not allowed to say lipids are polymers of fatty acids, because there's not just one single fatty acid that we can form together all the time. Next thing is that unsaturated fats are broken into two categories: cis or trans. Cis being that they're on. Cis is just another word for same. Trans is opposite. And so for cis, we're referring to the hydrogen groups. And so if you look on this picture right here, this would be considered a trans bond, a trans double bond, because we have a hydrogen on one side and then the other on the other side. Now, if we were looking at cis, then we would see that the hydrogen groups are on the same side. And so that's why we call this a cis double bond. Now, the importance of that is that when we're talking about trans, if you compare these two structures of saturated and trans, you see that they're almost linear. They're almost both linear. Saturated is pretty much linear, even though it's like a zigzag line. It's pretty linear in its shape. It's pretty straight. And then trans is also a pretty straight chain. But when we look at cis, it has more of a bend to it. It's more like this instead of being very straight. And that's going to affect how tightly we can put a bunch of molecules together. So for this one, because it's basically linear, you can put one right here. You can put one really close to the saturated with another saturated and another saturated and another saturated. And it could be very condensed. But when we look at cis, they have all these bents. They're all bent, so they can't be put that close to each other. So that affects what they are at room temperature. So for this one, this would be solid because they're allowed to be so close to each other. And also trans could be solid at room temperature because they're allowed to be so close to each other. And then cis, they would be liquid because they're not allowed to get so close to each other. And so they would be more spread out. And so that's why they're liquid. Last thing is that in the addition to glycerol, which is, yeah, that's not, you can't see that. There. So glycerol is this 3-carbon molecule. There's hydrogens and all that other stuff. 3-carbon molecule that has hydroxyl groups. That's where this O come from. But basically, when you have this molecule, you can put at least 3 fatty acids or you can add at least 3 fatty acids to this molecule. You can add 3 fatty acids to this molecule, and then what's it called? It could be any 3 fatty acids. It could be a saturated one here, an unsaturated one here, and then it could be another saturated, and then that saturated one could be 40 carbons in length. This unsaturated could be 20, and this one could be 5. Like, it could be anything. It doesn't have to be all the same. But once you add them to the glycerol, through the process of dehydration, because this OH group will interact with this OH group, and you'll take out a H2O to put this fatty acid molecule close to H2 or close to glycerol. Can you see that? No, that's not now. All right. So once we have a glycerol molecule attached to 3, that's what we call a triglyceride, because we have 3 different fatty acids attached to it. Now, or triglyceride, or what we usually call, a lipid. Now, the next thing is, if we take one of these saturated fats, or we take out one of these fatty acids, and we put a phosphate group instead, that's what we call a phospholipid, because we have a phosphate group with 2 fatty acids. And in case of that, this molecule would be called amphipathic. Amphipathic. Because it has a polar region, the phosphate group is very polar, and it has non-polar regions in the fatty acids. And so that's important because it plays a very key role in membrane structure. But besides that, that's about it. The last thing I would say is that this bond right here between the carbon, between the glycerol and the fatty acid is called a ester linkage. Yeah, an ester linkage. Basically, it's an O connected to a carboxylic group connected to a carbon on both sides. But yeah, I think that's about it. That's all you need to know for lipids. Just review how this amphipathic behavior allows it to become a membrane. Polar heads face outwards to the water, and then the hydrophobic tails will want to come closer together, and so you can have this double structure right here where these are the polar heads, and they form a phosphate bilayer. And that's about it. But yeah, that's all you need to know, I think, about lipids. Also, make sure you don't forget this, that they're not a polymer, because it's a very dumb question to lose points on, especially on the final. But yeah, that's about it. Yeah, so yeah. Okay. So we're out of time. This video will be posted right after, will be posted after the session is done. And if you guys still have questions, Ben will have a Q&A soon.
Closer to your final.
Close to the final. Look out on Canvas for announcements for that. Lastly, I'm going to post an announcement on the Canvas page for this class to talk about, or to link you guys to a survey that I created for the online CSS. It's a really short survey. It's anonymous, so so you guys can tell us all the good things you like about this, or all the bad things that you think we can improve on. And yeah, I had a great semester doing this. I'm sure I speak for Derek and David as well. So I guess that'll be it. Like I said, if you have any questions, Ben will post a Q&A closer to your final. So look out for that. And feel free to check out the survey. I guess that's about it, right?
I think that's about it.
All
For the
right.
last time,
See you
signing
guys.
off.
See you guys.
Annyeonghaseyo.